give you a warning at 15 minutes. And it'd be nice if you can finish a little earlier, then we can have questions. So I'll give you the 5-minute warning at 15, and then a 2-minute warning at 18. But I would like you to finish before 20 minutes.
Yeah, sure. Okay. Around, I think I can finish around 17.
Okay, perfect. All right. Thank you for sticking around. We have an exciting bunch of talks and proceeding presentations. And so this is one of them, one of the proceedings. Hao will present DIFFUSE, predicting isoform functions from sequences and expression profiles via deep learning.
Thank you. Okay. Hello, everyone. I'm Hao. I'm a PhD student in Professor Tao Jiang's lab at the University of California, Riverside. I will be presenting our paper on DIFFUSE, which is a deep learning-based method for predicting isoform functions from their sequences and expression profiles. So I'm sure a lot of you are familiar with the central dogma, which states how the genetic information is passed into proteins from genes by transcription and translation. Between the transcription and the translation, there is a mechanism called the alternative splicing. In this mechanism, gene exons are selectively included in mRNAs, which results in the generation of multiple protein isoforms from a single gene. So you may have listened to several talks about the study of gene functions, but in fact, these isoforms instead of genes are actual function carriers. So the goal of our work is to study the functions of isoforms. So first of all, studying isoform function is very important. The expression of different isoforms may lead to diseases. For example, the expression of the longer isoform of the TTN gene makes a muscle heart weakened compared to the normal heart. Other isoform-related diseases include cancers or the Alzheimer's disease. So understanding isoform functions can lead to or deep understanding of these diseases. On the other hand, gene functions have been well studied and recorded. One of the most popular databases is the Gene-Ontology database, or GO. In GO, each function is represented as a GO term, and each GO term belongs to one of 3 main categories. All the GO terms are organized as a directed acyclic graph, or DAG. In the DAG, each node is a GO term, and the edges indicate how more general GO terms is divided into some more specific functional terms. So with these defined terms, the GO database can describe gene functions by tagging each gene with a set of GO terms. So even though the gene functions have been well recorded, the specific functions of most isoforms are poorly understood yet. So some specific examples have shown that isoforms from the same gene may carry distinct or even opposite functions, but we don't have such a database that comprehensively record isoform functions. So that's why we would like to develop a computational method to predict isoform functions by integrating the data of isoform-level biological features and the gene-level functional annotations. That is, we want to correctly distribute the functional annotations of a gene to its isoforms according to isoforms features. And formally, if we treat each GO annotation as a binary label, we can treat the prediction of each GO term as a binary classification task. And we have some constraints on the labels. If a gene is labeled as having a function, then we know that at least one of its isoforms should have this function. if a gene is labeled as not having a function, then none of its isoforms has its function. So the nature of this problem is a multiple instance learning problem, in which each gene is a bag and its isoforms are instances in the bag. So if a positive bag contains at least one positive instance and a negative bag contains no positive instance. So several methods have been proposed for this problem in recent years under the multiple instance learning framework. The limitation of these methods is predicting isoform functions from the information contained in the expression profiles alone. So in our work, we propose to integrate more functional related data, including isoform sequences and conserved domains. Isoform sequences contain the information of short functional sites or motifs. Conserved domains are longer part of the sequence, which is conserved during the evolutionary process, which may imply their importance in functions. So we develop a deep learning model, DIFFUSE, to integrate various kinds of data and train this model without the isoform level ground truth labels. So basically, our model goes through two stages. In the first stage, we use a deep neural network to capture two levels of sequence features and output the likelihood of having a function for each isoform. We name the output of the DNN as initial scores. Then in the next stage, we use a conditional random field to combine initial scores and co-expression relationships to refine the predictions. And we jointly train these two components under the semi-supervised learning fashion. So more specifically, in the first stage, two levels of sequence features are extracted from the isoform sequences. First, each isoform sequence is represented as a series of overlapping trigrams, which are fed into the convolutional component to detect their functional motifs or functional sites. On the other hand, domains are extracted from the isoform sequences and are fed into the long short-term memory component to detect their order information. Then the initial prediction is made from the concatenation of these two features. In the second stage, we first derive the co-expression networks from the RNA-seq data. Isoforms with similar functions are often co-expressed. So we want to assign the same label to the highly co-expressed isoforms. At the same time, we want the label to be consistent with the output of the DNN in the first stage. So that's why we design such a Gibbs energy as the objective of our CRF. So this objective contains two terms. The first term is the unipotential, which penalizes the label assignment that is different from the output of the DNN. The second term is the pairwise potential, which penalizes different labels assigned to the highly co-expressed isoforms. And we have parameters to control the relative importance of these two terms. And we use a mean field approximation to approximate the likelihood of each label assignment iteratively. So as aforementioned, we don't have the isoform level ground truth label, so the traditional semi-supervised learning algorithm are not applicable to train our model. So we propose a semi-supervised learning algorithm to train this model with only the gene level ground truth labels. So here we first initialize instance labels as their bags labels. So in the positive bags, each instance is labeled as positive, and in the negative bags, each instance is labeled as negative initially. Then after making the prediction for each isoform. We can compute the loss using the current labels and update the model parameters of the DNA and the CRF using the common gradient methods. Then we fix model parameters and make inference for each instance again. And according to the inference results, we update instance labels under the MIL constraints by making sure that there's at least one positive instance in the positive bag and keep the instance labels in negative bags unchanged because they are all always negative. Then with the new labels, we can train the model parameters again, and we alternate the model parameter updating and the instance label updating, these two steps, until reach the convergence. And because we don't have the isoform level ground truth labels, we cannot perform the instance level or isoform level evaluation directly. But the maximum prediction score among isoforms of each gene can indicate how likely does this gene possess the label. So we check its consistency with the gene's annotation ground truth. So we perform both AUC and AUPRC to evaluate the performance. We compared our method with 4 aforementioned methods on 3 data sets. And the table shows the average AUC and AUPRC for all the methods across 3 data sets. And our results achieve much higher accuracy, much more improvement than the existing methods. However, this evaluation didn't actually validate the correctness of predicted isoform functions. Right? Because think about if all the isoforms of a gene are predicted as having the same functions. So actually, this is a main challenge of isoform function prediction problem. To address this challenge, we did several indirect validations. So we first test how the similarity of predicted functions is correlated with sequence similarity or expression similarity, because we expect that isoforms with similar predicted functions has the similar sequences or similar expression profiles. So we group all the isoforms into clusters and estimate the average functional sequence and expression similarities within each cluster. Then we use the Pearson correlation coefficient to measure the correlation between functional similarity and sequence similarity, and so on. The results clearly show that isoforms within groups with higher sequence similarity or expression similarity tend to have more similar functions. Interestingly, we found moderate correlation between the sequence similarity and the expression similarity, which may imply why these two kinds of features can be combined to improve the functional prediction. In addition, some study has shown that protein structures are more conserved than sequences, which may imply that isoforms with similar functions could have more similar structures than sequences. To test this, we collected contact maps to represent isoform structures and did the same experiment as above to measure the correlation between functional similarity and the structural similarity, as well as the correlation between functional similarity and sequence similarity. As expected, we found higher correlation between the functional similarity and the structural similarity using our prediction results. This is another indirect validation we did. The UniProt database defines a set of functional sequence features, such as Mendel ion binding site or the nuclear localization signal. The presence or absence of the functional sequence features can be used to infer the potential isoform functions. For example, there are 4 Mendel ion binding sites in the red axon, and there is 1 nuclear localization signal in the green axon. And because isoform 1 sequence contains the red axon and isoform 2 sequence contains the green axon, we may infer that isoform 1 has a function of binding Mendel ions, and isoform 2 is located in the cell nucleus. So we find 3 sequence features that can be clearly mapped to geoterms, and check the consistency between the presence or absence of a kind of sequence feature in isoforms and the prediction results on the corresponding geoterm. We use the J-card index to measure the consistency. So this is the results for one of 3 features, the nuclear localization signal. Our results achieve the consistency with the sequence features of score 0.7 in terms of the J-card index, which is much higher than other methods. To make a summarize, we have developed very effective deep learning-based methods for predicting isoform functions from the gene functions and isoform sequences and expression profiles. And there is some room for improvement in the future. First, in this work, we have used the contact maps to represent structures to validate our predicted functions. But in the future, maybe we could incorporate contact maps in our model as the input feature to more accurately predict isoform functions. In addition, we can apply our methods to predict other types of functions, such as pathways or interaction with non-coding RNAs. From the method's perspective, we borrowed the idea of formulating the CRF as a layer of DNA to enable the fully end-to-end training, and the new method of evaluation and validation should be explored to better measure the prediction accuracy at the isoform level. And lastly, I would give thanks to everyone who's helped me with this project, especially Professor Tao Jiang, who is my advisor, and Professor Jianyang Zheng, Professor Dongbo Bu, as well as my colleague, Dipan Shao. They give me a lot of support and help along the way. And finally, thanks to everyone for coming to listen to my presentation. Thank you very much. Thank you.
Questions. The first one is, you mentioned there's no ground truth for function assignments for splicing, but within the Go consortium, we occasionally assign functions of different isoforms. But you may not have been aware of that, so I was wondering if you were aware of that if you were planning on using it.
Yeah, you have some small scale ground truth for the isoforms.
Yeah. It's probably not enough for you. We've probably under annotate that, but it does exist.
Yeah, that's good to know. We can use this small data set to do the validation, maybe. Yeah.
And then the other questions may be harder, but there's ongoing debate about, to what extent is alternate splicing, is it just noise, or does it?
I see.
Is that something that you've looked at, and your results kind of add to that debate?
Yeah, but yeah, right. Maybe there are some alternative splicing on noise, so they generate the isoforms with no functions, right? But some studies have found there are a lot of isoforms of the same gene has different functions and maybe lead to different diseases, like the TP53. They have like the different isoforms from the same gene may have different functions, like the apoptosis, the positive regulation of apoptosis or negative regulation of apoptosis. So there should be a lot of isoforms that are functional. So I think this prediction is meaningful. Thank you.
We have time for one more question, please. Okay, if not, let's thank our speaker.
Thank you.